now. So
I've got it figured out. I just had to reload it from scratch.
wonderful.
So I got fascinated by mushrooms in graduate school. It was 1968. Somebody gave me a bag of morels. Prior to that, I didn't like eating mushrooms. I never hunted mushrooms. I had no interest in pursuing biology. And that bag of morels changed my life. And now we'll talk about some of the things that have happened since then. And first of all, I want to give a clear definition of what the Ascomycota is. And that's really what I should be using as a term, because Ascomycetes is a lower classification than the Ascomycota. So we've got two general broad groups of fungi in the fungal kingdom. The Basidiomycota, in the upper left, you see a scanning electron micrograph taken by Paul Stamets of a single basidium with its 4 basidiospores, the mature ones in the foreground, and there's some more immature basidia in the background. And in Basidiomycetes, the spores are released directly from those basidia. But in Ascomycetes, the spores are grown and created inside of a sac called an ascus. Now, the sac is typically, most of the time, a long, narrow sac that has 8 spores lined up end to end, but it can be a round sac, like is in tuber, with just 4 spores, or it can be a sac inside of a big sac, and there can be up to 100 or even more than 100 spores inside some of those particular Ascomycetes. And the ones that have the spores lined up end to end in the sac have 2 different ways of releasing their spores. The one group are called the operculate Ascomycetes, and these are the Pizizomycetes, and those are the first things we're going to look at. And the upper left and the lower left image shows an operculum opening and releasing the spores. So when the spores are mature, that lid on the top opens and the spores squirt out. Now, an inoperculate Ascomycete, the image on the right, has a pore at the very tip. And when the spores are mature, they get spit out, kind of like you would spit out a watermelon seed. So it's a very different thing. And there's a whole bunch of different classes of Ascomycota that are inoperculate Ascomycetes. Now, but if the spores are born in a sac, either operculate or inoperculate, most of the fungi that you will identify and pick that are in the Ascomycota produce those spores in what's called an apothecium. In other words, the spore sacs are lined up side by side in parallel and then release the spores when they're ready. But there's also parathecial ones where the spores are inside of this kind of pear-shaped structure, but inside of the pear-shaped structure are the individual asci, and those are also inoperculate fungi, i.e. when the spores are mature, they get squeezed out through that little hole. Now, there's also a lot of microscopic fungi that I will not talk about that are produced in a clysothecium. And those are the ones where you can have not just 8 spores, but up to 100 or more spores inside the sac, inside of a larger sac. But I'm going to be talking about apothecial and parathecial fungi. Now, the other thing that's interesting about these Ascomycota is we can have both an asexual phase and a sexual phase. Now, the asexual phase, for example, of a Pizizom is the chromelosporum, and those are the image shown above, and you see an actual fruit body. But most asexual phases of the Ascomycota are microscopic. You've got to look at a microscope in order to see them. And some common ones that you'll see every day is like penicillin mold on bread or on your rotting orange. That's the green that you see as these little round conidia, which are asexual spores that are produced in a phyllide on the penicillium mold, or fusarium, which is a wilt, produces a very long asexual spore, long and narrow, but also produced on what's called a phyllide. These asexual forms, but what as foragers, and I'm mainly a forager, are interested in is the sexual forms. And this is Coreactus giastra, and it's found amazingly in Texas and northern Japan and nowhere else in the world. So, and it's a Pizizomycete. And the first half of this program is going to deal with the Pizizomycetes. And if you tap the side of one of these Pizizomycetes, when it's mature, in about 5 seconds, you'll get a cloud of spores. And so this first photo shows the spore discharge of an apothecial member of the Ascomycota, and particularly the devil's cigar. Now as a harder spore discharge to see. This is Sarcosphaera coronaria. It's another Pizizomycete, as the next about 20 images are going to involve. And Sarcosphaera coronaria is an edible mushroom. Orson Miller years ago told me it has the delightful flavor of eating a pencil and texture of eating a pencil eraser. And it also is a hyperaccumulator of arsenic. And it's something to think about when you're collecting fungi. Always collect fungi from good environments because they can concentrate chemicals from the surroundings.
Mr. Beug, may I
Yes.
interrupt with a question right here? You said this was a hyperaccumulator of arsenic. Is there a
Yes.
specific reason why this fungus is attracted to arsenic more than others?
Don't know. Morels are hyperaccumulators of gold, and I don't know why that either. But some fungi, like morels, if there's lead in the soil, morels will have lead in them. If there's arsenic in the soil, morels will have arsenic in them, but they'll have less arsenic and less lead than is in the soil itself. A hyperaccumulator has more lead or more arsenic than in the surrounding soils. And that's the distinction. And for some reason, Sarcosphaera, the tulip cup, is a hyperaccumulator. And I find that fascinating, but an important thing to know, if you're going to eat certain fungi, be extremely careful about where it's growing. That's the important message. So now I want to move into the details of how we identify some of the popular edibles, like Verpa bohemica. And for a long time, it was proposed that this should be moved to Tychoverpa bohemica, but that name never really came on. But these things are called early morels, but they're distinct from morels in that the fertile area, that wrinkled structure at the top, hangs like a skirt from the very top of the cap, and the stipe goes all the way up to the very tip top. And also in Verpa, the stipe is typically filled with a cottony material. And around in the Northwest where I live, and probably throughout the country, these tend to fruit when the new leaves are about the size of mouse ears. And they like river bottom areas, so they'll be under cottonwoods and poplars in the spring. Interestingly, the color isn't very useful in telling one from another. I found this beautiful apricot orange Verpa one day, and I said, oh, I was sure I'd found a new species, but DNA showed it just to be plain old Verpa bohemica. Now, morels, in contrast, and you can see it in just one image in the upper right, when you've cut it in half, the stipe doesn't go all the way to the top of the cap. The stipe is attached at the bottom. And I greatly prefer eating the true morels to eating Verpas. The Verpas are edible. They have a reputation of being poisonous because some people have a bad reaction to them. But about an equal percentage of people have a bad reaction to morels, where morels are so delicious that we call those edible, and some people call the verpas poisonous. But they're both equally edible, but both must be thoroughly cooked. Now, this first morel I'm showing here is the landscape morel. In my book, it appears as Morchella elata, and that was the plant. When I actually wrote the Ascomycete fungi of North America, these morels did not yet have a name. It was just in the process of being settled. And I set my entire manuscript, the chapter on morels with all the photographs, to the team that was working in Europe. And they told me what name to apply to what morel. And but they changed their mind. They were going to call this Morchella elata. Right now, there is no Morchella elata, because if you read the Frisian description carefully, that's probably was a burn morel and not a landscape morel. And so they abandoned that name and went to Morchella importuna. But be aware that there's many different morels that can grow in landscape areas. So on the very top left, there's an image that I thought when I took it was all Morchella importuna, but I may have as many as 3 different morels in that one photograph, all growing in the same landscape bed. And only the one on the far right is importuna itself. And it's a particularly distinctive black morel or elatoclade morel, in that the vertical ridges are very, very close together, and there's an abundance of cross ridges that give this a very distinctive ladder-like appearance. And now here are the burn morels by contrast. And if we take a look at the picture in the upper left, that could be any one of 4 different species: Morchella eximia, Morchella exuberans, Morchella sextulata, or Morchella septimulata. You need DNA to tell them apart. Now, if you had asked me how many indistinguishable burn morels there are 3 months ago, I would have told you just 3. And so we're constantly learning new things. Now, the image in the upper right, I actually went in, I had sent it to France at the time they were doing the paper. And so I do have a DNA sequence for that. And it turns out to be Morchella eximia. So but the important thing to note is it could just as well be eximia in the image on the left. In fact, it probably is. So the color of the pits is not at all reliable in morels, but it's the color of the ridges that's important. So even in the young Morchella eximia, the pink one, you have a black ridge, and it'll get blacker in age. Now down below, we have the gray morel, Morchella tomentosa. This morel is another burn morel, but it's actually not related to what we typically call the black or the blonde morels. It's in its own separate clade. It grows from a sclerotium in the ground, and it grows in the fire area, but it grows after all the other morels are just finished fruiting or totally finished fruiting. And if the weather conditions are right, Morchella tomentosa can keep fruiting right up until snowfall in October. So this, if you think about morels as something you just go hunting in the spring, if you know about Morchella tomentosa, you can actually stretch your morel hunting all season long just by following the areas where there'd been a burn a year or 2. And sometimes the Morchella tomentosa will fruit 3 or 4 or 5 years after a burn, where most other burn morels primarily fruit 1 year after a burn, unless it's been dry, and then they'll fruit more than a year after. Now, the most common eastern morel was long thought to be Morchella angusticeps. In fact, that was basically the only black morel that was known from the east. So if you found a black morel on the East Coast, you had angusticeps. Now, a few years ago, I sent in a morel sample that I found in Washington State to the same DNA person, Kerry O'Donnell, that Andras Wojcik's sent his morel in for analysis, and his morel came from Newfoundland, and they were identical. And after considerable thought, and we decided to name this thing Morchella eohespera. Eos for the goddess of the sunrise, and Hespera for the Hesperides of the sunset, because it was found both in the east of North America and in the west. But it turns out it's the most common morel in the Alps, in Siberia. It's the most common morel throughout North America, the most common black morel. And yet when Michael Koa was doing his 10-year study of morels of North America, this one didn't show up. And when Cloez did his morels of the world, he missed this one completely. But after we got Eohespera published, an old collection from Norway showed up, and that turned out to be the same as ours. And that collection had been called Morchella norvegiensis. So the first name wins, although I love the name Eohespera because it properly gives you the range of the mushroom. Now, this mushroom grows under hardwoods, under conifers. It grows at sea level. It grows at 4,000 feet. It's everywhere. And the other thing to know about morels that most people don't realize, their asexual phase, at least of many of the morels, is an endophyte on grass, something you don't see at all. But in between seasons, that's how the morel is passing its time as an asexual endophyte on the surface of grass. I should title this slide, these are the half-free morels. We long thought we only had one species, and we called it Morchella semilibera, but we now realize the East Coast species, Morchella punctipes, is different from the West Coast species. But this year, I've come to realize that we have at least two and possibly three different things that are all passing as Morchella populophylla and look very different and actually taste very different. My favorite is the one on the top left. It's a huge, meaty thing. I've only found it once, and we didn't have a good idea whether it was really good to eat or not, but I'll assure you it's delicious. But right now I'm busy trying to figure out which of these things is the true populophylla and which one is going to need a new name. Now, Morchella americana was named Morchella esculentoides by Kuo in his big study, but the Cloez paper was published just like a few months before the Cloez paper, and Cloez had called this mushroom Morchella americana. Now, he had called color varieties of Morchella americana a half a dozen other species. And all those names got thrown out because they turned out to be identical to americana, because the pits can be a range of colors from almost black to this very, very blonde morel. But it's the genetics that you're going to need to be able to run to tell exactly which blonde morel you have, or the esculenta clade morels. And we have Morchella americana throughout North America. It is coast to coast. But if on the East Coast, there's a number of similar morels that are in the blonde morel group, or the esculenta clade, that are not found in the West. And that would be Morchella diminutiva, septiformis, and ulmaria. Now, I have a CF on the first few because those photographs were all taken before DNA. So I can't be positive exactly what morel was photographed. But the Morchella ulmaria by René Leboeuf actually is sequenced. So we know that even though it looks just identical to americana, it is a distinct species. And the top two mushrooms in this image were known as Morchella dunensis at the time I was writing Ascomycete of North America. And then I found a morel in actually a friend found it and invited me over to her home to see it, that we sent to Kerry O'Donnell, and he said it was Morchella vulgaris. And we included that in a paper we were publishing. I was publishing with a team. We were publishing some Central and South American morels and one from the Grand Canyon. And we had said, oh, we have Morchella vulgaris in North America. And the Europeans said, no, you've got Morchella dunensis. But since then, we now know those are identical things. But they're very similar to a morel that is definitely in the West and probably in the East as well, Morchella prava. They look sort of like a contorted Morchella americana. And one of the distinctive features is the ridges are very wide on both Morchella vulgaris and Morchella prava. And now we'll move into the genus Gyrometra. Right now, this genus is in a huge state of rebellion, and we can expect to see a massive number of name changes and new species in the coming few years. There's a big study going on at the University of Michigan. I've sent all of my herbarium material on Gyrometra from 1999 to the present there for DNA sequencing, and we're finding all kinds of interesting things. So the first three mushrooms are in the genus Gyrometra, and the fourth one is Dicyotis venosa. Now you notice I have the word group after that because there's actually only one. It's a morel in the related to morels, but I found this year alone 4 additional species in addition to the one in the photograph that match Dicyotis. So this thing is going to be turning into from one species into a minimum of 5 new species in the relative new future. Gyrometra leucoxantha, we can expect to see some changes. Gyrometra ancellus and leucoxantha were once in the genus Dicyna, and all of the Dicynas were thought to be free of gyrometrin. And Alden Dirks at the University of Michigan has recently found Gyrometra leucoxantha has gyrometrin in it as well. And because gyrometrin is potentially deadly and is definitely carcinogenic, it's now advised that you don't eat any of these little flat morels. Dicyotis venosa is safe enough if thoroughly cooked. You just have to be able to distinguish it from the Gyrometras. And Gyrometra melaleucoides is actually very unrelated to the top 2 Gyrometras. It's at the very basal clade. This is what Gyrometras originally look like. And all the diversity in the genus Gyrometra comes from the evolution of relatives of Gyrometra melaleucoides. And we tell these things apart by looking at the mature spores. So if you want to study any of the Ascomycota, it's really important that you get a fresh spore print and that you take it. I actually spore print my mushrooms directly on the glass slide, and that you use either amount of water or amount of Lugols, and never mount them in Meltzer's or in KOH, because that can change the features of the spore itself. But we tell them apart, like Gyrometra ancellus spores are kind of thin, but they have these little points at both ends. And Gyrometra leucoxantha has the spores like the ones on the lower left, but they shouldn't have been as rough as the spores I was looking at. So I thought I might have a new species, because I had been trying to figure out this one in the middle, which is right now called Gyrometra spanuv, a new species. And it was sort of a pinkish Gyrometra leucoxantha, but it turns out it's a distinctly separate species from leucoxantha, but also it has a little bit of gyrometrin in it. You see, the melaleucoides spores are much smaller and they're unornamented, and the Dicyotis spores are typical of morel spores, these large things without an oil drop inside, but often with a little tiny oil drop somewhere on the periphery, on the outside of the spore. So the spore print is going to be really, really important in getting your identification if you decide you want to study the Ascomycota, and getting it right, i.e. taking fresh spore prints. Once the mushroom is dried and in the herbarium, the nature, the appearance of the spores change. So you need to know what they look like fresh. So here's some more gyrometras. And at one point, Gyrometra montana in the lower right and Gyrometra corfii in the upper left were both considered synonyms of the European species Gyrometra gigas. So when I was writing the Ascomycota book, I called Korf, who was still living at that time, an incredible mycologist at Harvard, and he said that Corfii was named not in his honor, but for scolding him by it was a European mycologist who said, "Hey, this is not the same as gigas. You shouldn't be calling it the same thing." So this is a name based on a scold. But all of these mushrooms that you see, we're likely to soon be discovering are species complexes. So we probably have more than just Gyrometra brunea out there. We definitely have at least 2 species that are Gyrometra montana. At least I'm pretty convinced of it. And we probably have multiple species that'll match Gyrometra corfii. And all of that is a research project of Dr. Miller at the University of Michigan right now. And Gyrometra caroliniana, the red morel, it can reach the weight of a pound or 2. It's quite a spectacular morel. I've never seen many of these mushrooms myself, but in writing the Ascomycota book, I wrote to every photographer in the country, and everyone shared all of their photographs. And the way I chose the mushrooms to include in the Ascomycetes of North America book was every photograph, every species that anyone had a photograph of wound up in the book. If I couldn't get a photograph, it wasn't in the book. If I could get a photograph, it was. And so it wound up with around 550 species in the Ascomycete book.
Michael, can I jump in
Yes.
and ask a question? In your previous slide that you were just showcasing, you had the cross section towards the bottom right corner. In that cross section, it displays the chambers located inside of the Gyrometra
Montana. The same chambers inside of Gyrometra corfii, by the way.
Right. And so these chambers, when I saw that, instantly reminded me of some hypogeous fungi. I forget the name of it right now. Geopora.
We'll see one in a few minutes.
Yeah, the geopora. So are these direct relatives that have formed a stipe or a stem?
No. What you have is what we call convergent evolution, things that are unrelated that evolve the same form but aren't related. And you can also have things that look very different that are related. So it's just an evolutionary quirk.
Oh, okay, got you. Thank you for clarifying that.
Yeah. So if you had asked me in June, I would say we have Gyrometra esculenta in North America, but I had trouble explaining why there's a lot of deaths from Europe, but so far no deaths from North America from Gyrometra esculenta. And it turns out we don't have Gyrometra esculenta at all in North America. At least it hasn't been found yet. I can't say for sure we don't have it. So these color forms that you see here might represent different species. I know, based on what Alden Dirks has told me from the University of Michigan, that we've got at least 2 species in the Pacific Northwest and even more species in the rest of North America. So Gyrometra esculenta is a species complex or a group. And some of the ones we have in North America have relatively little gyrometrin and are not too likely to cause liver damage. Some of them have very high levels of gyrometra and are very likely to cause liver damage, but they all have at least some gyrometrin and are all potentially carcinogenic. So even though some people love the flavor of the Gyrometra esculenta complex, I say don't eat them. Now, I'm also puzzled about Gyrometra ambigua versus Gyrometra infula. And because sometimes I find a little tiny Gyrometra thing that exactly seems to match spore-wise with Gyrometra influella versus the big one on the right. And now we begin to realize these probably are two different species. And earlier this evening, I was in contact with Alden Dirks and in the University of Michigan and Danny Miller up in Seattle to try and straighten out what's going on with Gyrometra ambigua. We have probably several species. Interestingly, these are all three considered to contain gyrometrin, but so far Alden has not detected gyrometrin in any of these. I'm not going to start eating them, however. Now we'll switch over to Helvella, and there's sort of the Helvellas that have a stipe or a stalk, I divide them kind of grossly into the ones where the stipe is lacunose or folded or ridged versus ones that have a round or a terite stipe or stalk. Now, the one where the stipe is totally buried and you don't see it till you dig it out has been put in a new genus. It's now Dicingia leucomeliana, very, very common mushroom. Most of these mushrooms here in this image are found throughout North America. And Helvella crispa is widespread. Helvella sulcata is an eastern species. Helvella vespertina is a western species. This spring, I found at least 2 yet unnamed Helvellas that fit into the broad definition of these crispa vespertina group. And so this is another area where there's going to be some new species showing up soon. At least Helvella vespertina, if you cook it thoroughly, has a very morel-like flavor, but it's very, very rubbery in texture. But it is edible. These other round terite Helvellas tend to be real small, of not tempting to try for food. They're separated largely by spore size and shape, by the color of the underside of the cap, and by whether the underside of the cap is furry or not. But I just find them beautiful and intriguing to look at. And but we've seen a lot of name changes, and we're soon going to be seeing a lot more. Now, if you want to really get into a mess, start trying to identify Pizizas. Piziza varia can be just about any color you can imagine. The spores, even in the same collection, change shape and size over time. It makes it an incredibly difficult mushroom to identify. It can look exactly like Piziza domiciliana, which is famous for growing in wet basements and shower stalls and on an old chair left to mold. And but it could equally be Piziza varia. It'll grow in the same habitat. Piziza vesiculosa is one of the few Pizizas that's relatively easy to identify because it tends to grow on old straw, particularly a manured straw. And this one has been a little bit fried on the margin, but the edge of the margin of the cap tends to be neatly sculpted. Piziza arenaria is a nice eastern Piziza. Now this spring I found a beautiful purple Piziza that exact match to a European species. Everyone says it's not in North America, but I'm pretty sure that's what I found. It's out for DNA, but there's a lot of interesting Pizizas out there. And again, more changes in nomenclature. Piziza sublilacina, as it appeared in the book, because Piziza violacea was judged again by Korf as a nomen dubium et confusum, because some people considered the smooth spored purple burned mushroom, and the Pisiza, and this mushroom is associated with burns. The smooth spored was Violacea, and other people know said, no, it's the one that has the ornamented spores, which we now are calling Pisiza praedavisa. But now when it got epitypified, because a European wanted to resurrect the name Violacea, so he epitypified the burn morels, that the burn cup fungus as Geoscypha violacea, and it has smooth spores. And there's a lot of really gorgeous little cup fungi out there. Cocainus sulcispes is basically a tropical species, but you'll find it in the southern and the warmer eastern states. Sarabiella renana is one of the snow mushrooms. It grows from a sclerotia. And when it's young, you'll just get 2 or 3 or 4 of these cups. And the older, year after year, you'll get more and more these cups in the fruiting. I've never been fortunate enough to find it, but it's a very, very beautiful thing. So I just love the beauty of these things. And then there's Caloscypha fulgent. All 3 images are the same mushroom, even though the color is very, very different. The one in the upper left is an albino form that doesn't generate any color at all. And so the green stain you see on the outside of typical Caloscypha fulgent is actually robin's egg blue, but blue on orange to yellow is a green color. And so here's Caloscypha fulgens, a seed parasite. It's a Western species, but it's also very, very common in Colorado, which is probably as close to Texas as you'll find it. Melastesia chatterai is another very interesting mushroom. It has fine black hairs all on the margin of the cup. Makes it very beautiful to look at under a hand lens. Aleria aurantia, the orange peel fungus, very widespread. The 2 Sarcoscypha species are found strictly in the eastern North America. We have a lookalike in the west, but it's a different species. And Plectania melastoma, we now realize is 2 different species, is found all over North America. And gorgeous thing. And these pseudoplectanias and are also very interesting. Just they're hard to spot because they're black, and so you often miss them. And then there's the little cup fungi that are like Otidia nautica, Otidia unicisa, Winnie americana. But many of the Otidia species just are very, very difficult to identify, and so remain unnamed. And among the my favorite little tiny mushrooms are the eyelash cups, Scutellinia scutellata, which I thought was easy to identify until this year, and now I've had to add the word group to it. And so if I see a Scutellinia, I just call it an eyelash cup and stop there. It gets very hard to tell exactly which one you have, but they're gorgeous and tiny. The ones in the upper left, although they're large, all 3 of them together would fit on my little fingernail. But there are things that have beautiful eyelashes on the outside that aren't related at all, like Kyllemania filmicola, which grows on manure. And Tuberlionia, which when it's mature has a wonderful odor, and it's an arumpet tuber. It's a tuber that appears on the surface of the ground at the time when the pecan harvest is ripe. And so the pecan farmers have learned that they have a crop growing with their pecans that's as valuable as or more valuable than the pecans themselves. Inemia gigantea is a large unrelated truffle that was found mainly, I think he first found it in the Smoky Mountains. But when it has a delightful odor, it has a delightful flavor. No odor, no flavor. The same with Tuberlionii. And now we talked earlier briefly about Geopora cooperi. This mushroom, you can carve off the outside. It doesn't have an aroma, but it has a very nice morel-like texture and flavor when cooked. Now, another truffle, Elaphomyces americanum, however, is not at all related to the Ascomycetes. So everything up to this point has been a Ascomycete. Here's the first example of a Mycete, and typical of Elaphomyces americana. We have an enormous number of species. Almost all of them are as yet unnamed. Now, two more, the last two of the Ascomycetes, Sarcosoma globosum, which until a year ago I thought was strictly an eastern species, but now it's been showing up in British Columbia and in the Olympics, so we know it's eastern and western. And Galliella rufa is also turning out to be far more widespread than we thought. Now I'm going to move on to the Sodariomycetes. These are the ones where the spores are in a sac enclosed in a parathecium. And I particularly like Cameropsis petersii, which has been called the dog's nose fungus because it's textured in the appearance much like a dog's nose. Entonoema liquescens is a southern species. It probably gets into at least parts of Texas. It has liquid inside, but it's another parathecial Ascomycota. Now, the hypocrias are all parathecial Ascomycotas. No interest is edibility, but of interest just in terms of their growth form. Hypomyces species are also parathecial Ascomycota. Hypomyces lactiflorum, I thought was almost exclusively on Russula brevipies, but recent DNA evidence has proven that it grows on several different Russula species. And sometimes people wind up getting ill after eating this mushroom, which is known as the lobster mushroom. And in those cases, I think it's been growing on a mildly toxic Russula, one of the hot Russulas rather than one of the brevipies group. And some of the brevipies group are somewhat hot.
That's interesting, Mr. Beug. I have a question here in the chat box from one of.
Yes. I need to figure out how to see the chat box again. I had it at one point, but it's disappeared. Well, anyway, tell me about it. What was the question? Well, anyway, I'll go on. If the question comes up, I'll try and answer it. Hypomyces lateritius is interesting in that it preserves the color of the Lactarius that it's growing on. So if it's growing on a member of the Deliciosis group, it'll be orange, but there'll be no gills because it's actually parasitized before it comes up. If I return to Lactiflorum for a minute, at first it's a gill-less mushroom that's all white because the initial stage, the initial spores that it's producing are white. And then it goes in a second growth stage where it's orange, and then it gets in the very fully mature form, it's really only good, and it's reddish purple, and it's at that point, it's a very good dye mushroom and not such a good eating mushroom. But Hypomyces chrysospermus, when it attacks boletes and eats boletes, and it just turns them into a stinking, soggy mess. There's nothing edible there at all. Cordyceps militaris is, and you can see the little parathecial all over the surface of the heads of that mushroom, and it parasitizes a moth larvae in the ground, or a beetle. I'm not sure exactly what it's growing on, but Hymostilbe sphingnum actually parasitizes and consumes the sphinx moth. And then Elaphocordyceps ophioglyphoides is actually an Elaphomycete truffle eater. And these are all parathecial Ascomycetes. There's quite a bit of interest in Cordyceps militaris as a medicinal mushroom. Most Ascomycetes, a lot of Ascomycetes are important in medicine, but those are all microscopic things. This is the only one of the Ascos that I'm aware of that there's medicinal interest in as a medicinal mushroom. And finally, Xylaria hypoxylon, Xylaria cubensis, this cognaxon, Annula hypoxylon. These are all parathecial Ascomycetes. And John Plischke's photograph shows both the asexual form, which is the white one, and the sexual form, which is the cubensis. And a few years ago, the decision was made that the asexual form had to have the same genus name as the sexual form. We haven't yet fully sorted out which wins, because it depended on whether the asexual form was the best known form, in which case the asexual name would take precedence, or whether the sexual form was the most known form, and then in that case the sexual name would take precedence. So these dual names for the sexual form and the asexual form are in the process of disappearing. Claviceps purpurea is the last Sodariomycete that I want to talk about, and it's famous as the source of lysergic acid, from which you make LSD, lysergic acid diethylamide hydrochloride. Now, Tophrina, which forms what's called alder tongues. Now, those tongues are not fungi. It's a misshapen growth of the cone itself, and the spores are just born naked on the surface. And so it's a tephrinomycete. It's a totally different group. And now we'll move on to the Leiotiomycetes, and the rest of the program until the very, very end will all be Leiotiomycetes. And these are all what we would call inoperculate Ascomycetes that are born on an apothecium. And some of them are really quite spectacular. Vibricium truncorum grows in streams, and you'll find it often just completely submerged, but in a real wet, boggy area, you'll see it. Spathularia flavida, another interesting mushroom. Mitrula elegans grows nationwide, and it's basically a bog mushroom. Sclerotinia sulcata grows from a sclerotium, very gorgeous. Noah shared this picture, which I used on the cover of the ASCO book. And in this photo, this is an interesting photograph to me. I was photographing the blue cheese polypore of Basidiomycete, and I carefully cut off the polypores and took them home. And it wasn't until I was processing my photographs at home that I realized that I had photographed 2 or possibly 3 different Ascomycetes. One, it's so young and small, is either Chloransuelia versiformis or Bulgaria inquinans, and the other is probably Melissia cinerea or Tapesia fusca. So the thing to realize about these ascos is some of them are quite easily missed, but can be quite beautiful and quite spectacular under a microscope. So here are some of these things, or they were intentionally photographed: the Chloransuelia torta, the Versiformis, Bulgaria inquinans, and the Melissia cinerea, or it could be Tapesia fusca. You can't tell just from looking. You're going to have to do microscopy. Leiotia lubrica, Neocudoneella and Ascotremella and Ascocorian are all these inoperculate Leiotiomycetes. I'm particularly fascinated by Neocudoneella radicella, and I pursued it for years because it grows on spruce rootlets in bogs. And I was in Wisconsin at the NAMA foray, and I got to go out with the mycologist Peter Kennedy, who I had known as a student at Evergreen College, along with the local botanist, and she led us to a fen, and we walked out onto a floating sphagnum bog. We took our boots off and forded out onto the sphagnum itself across a log, and we were walking on water 30 feet deep. And the sphagnum was so thick, and it was just glorious to walk on. It was a nice, warm, late summer day. And I was able to find Neocudoneella radicella growing from the spruce rootlets. And these are little things that if the weather's been dry at all, they'll just shrivel up and you won't see anything at all. So to get these photographs, they have to be in good moist condition where the Ascomycete has opened up and you can get a good look at it. What was long called Bispirella citrina in the last year has been reclassified as Calycina citrina. And now for one of the reasons this program has changed a lot, just in the last week, I found out that the geoglossomycetes are probably all actually lichenomycetes, which is a brand new group. And it contains fungi that live in the gut of insects, as well as things that look radically different than a typical Ascomycete, but all of them behave like lichens. In other words, they are symbiotes on other organisms. So Thwaimondidium atropurpureum, I know, is a lichenomycete from the paper, but I'm suspecting that Microglossum olivaceum and Brioglossum gracili are also lichenomycetes, i.e. organisms that depend on another organism for their existence. And we'll close with a neolectomycete. It looks similar to the club, but so unrelated that's in a totally different class. And that's my view of the Ascomycota.
All right. That's a fantastic presentation, Mr. Beug. I think everybody else will be clapping right now, but I have them muted for specific purposes. I think the one question I think I'm not sure if we got to it. How do tuberous species reproduce?
Well, they produce a fruiting body that produces spores.
Right.
And so the tuber is sort of like you think about a potato. You know, it grows this tuber at underground, and from it you can grow a new potato plant. You can take a tuberous species, plant it, grow it, you'll get the fruiting body that'll produce spores, and that's how it reproduces and spreads.
And how would somebody get a spore print from a tuber?
You don't. You grow it.
Right.
And when it fruits, you get the spore print from the fruiting body.
Got you.
Like a tuberous morel, you would spore print the morel that grew from the tuber.
And one of the other questions I have is, from my past experience hunting and burned out areas, I've realized that a lot of the pyrophilous fungi, the mushrooms that grow on burnt material, are actually Ascomycetes. Is there any specific correlation as to why basidiomycetes are largely absent in those areas?
I've observed exactly the same thing, but there's two things possible. One is a lot of the Basidiomycota that we're interested in are mycorrhizal. The burn killed the trees and killed the fung, and therefore the fungus hasn't got anything to feed on and reproduce. It may still be alive in the soil. In fact, it often is, but you won't see it again fruiting until a new tree has grown, and then it gets the nutrition it needs to reproduce. So there are Basidiomycetes that only fruit once every 10 years or once every 20 years. There's some of these things that I find on my driveway. I've found them once in the 20 years we've lived here. Some things I find every 5 years, but there's a lot of things that are widely spaced in their appearance. A lot of the things we pursue to eat are things that produce every year, but there's an awful lot of stuff out there that produces much more rarely. Like I think a chanterelle, the mycelium will survive, but it won't grow until it has new trees to feed it, and then it'll reproduce.
That's interesting. I have two more questions probably before I let you go, and I think everybody else they just really appreciate the presentation. The questions were more about evolution. Would you say that the Ascomycetes were broke off from Basidiomycetes, or which one was the ancestral fungus that originated from that?
I haven't got a clue on that
Yeah,
one.
so,
And I mean,
well, I figured I'd ask.
this is way, way back in time. I mean, when I started this, fungi were plants. And now we know that fungi are actually much more closely related to animals. They basically are at the base of the animal tree, not the base of the plant tree. So they evolved from our common ancestor. Now, whether the Basidiomycete or the Ascomycota evolved first, probably I don't even want to speculate. It's so long ago. And you see, the things aren't preserved. It gets real hard when we go way back because we need to find something preserved in amber or something preserved in some way that we can analyze it and know how long that species has existed or that variety or that type. We,
Thank you.
we can, by looking at DNA changes, tell how ancient the lineage is. In other words, we can tell what all the morels evolved from. A morel much like Morchella rufobrunnea. Everything evolved from that.
And my final question before I let you go, I thank you for your patience and wonderful presentation. What is the purpose of the ascus? Was that something that is just to contain the spores, or was that like another evolutionary trait that developed to protect the spores?
I view it as an evolutionary trait that protects the spores until they're ready. Now, the Basidiomycota have to use other tricks. Like in a king bolete, those tubes have a frosting that protects the spores until they're developed, or there'll be a partial veil, or there'll be a universal veil that'll protect those developing spores because they need to be able to develop this little water droplet, the Buehler's bubble, to release the spore. And if they dry out too soon, they can't release their spores. So they have to have some protective mechanisms. And where Ascomycetes have solved that problem by having the spores in the sac. So in one sense, you might say that the Ascomycetes are more advanced.
Yeah,
But I do not know.
Well, wow, mine is blown. I'm gonna have to process all of this information. I once again thank you for spending the time to share your knowledge with my club, the North Texas Mycological Association. We're all very happy to have had the honor of having you present to our club, and if we can have you come back in the future, we'd really appreciate it. Anytime, whether it's on Zoom or in person.
It's my pleasure. And you should know, I've got 25 free programs available through NAMA when you need a club presentation and your events fall
awesome.
through.
Yeah.
But nothing as up to date as what I just did for you guys, because this is up to date as of yesterday. I mean, I was learning new things.
And just having your interaction, it's just been awesome. Is sharing your knowledge, and you've been so helpful. And my whole goal was to share with my club your knowledge of Ascomycetes, and I think you've done a wonderful job of doing that, and I appreciate it. And
Okay.
I think everybody else has had a wonderful time. I will leave it at that, and thank you so much again. Happy New Year, Mr. Beug, and I hope to see you again in the future. Take care.
Well,
Bye.
I'm always here. Take care.
Bye. Take care.
Well, I have to assign a new leader in order to leave, so you need to take your leadership back if you could.